UM-Macau, and we are honored to be here for the first time to participate in the symposium with our friends in Hong Kong. This year it's our first time to choose the therapy track, and solving celiac disease role in intestinal microbiota is our object. Today we will introduce our project from those following aspects to give you guys an overview. First, doesn't like the lactose intolerance with really high prevalence. The CD is a chronic multi-organ autoimmune disease that has long-term effect in the region of small intestine with genetic reason. CD is worldwide food intolerance disease with 1% prevalence on average. In Western countries, the population of CD is significantly higher than our regions. One of the reasons why to choose this as our target is that based on the prevalence difference of CD between ours and the world, developing this new synthetic biology therapy can strengthen the ability of caring for different groups all over the world and status as the global tourist city of our Macau region. For celiac, the long-term impact with chronic diarrhea, constipation, etc. will influence life quality, and because the damage from the overactivated immune system, celiac's intestine cannot absorb the nutrition efficiently, causing the stunted growth of teenagers and children. According to the only two therapy ways, diet control and oral enzyme will have their inconvenience to patients and the immature of the technique. We try to find a way with higher efficiency and controllability to deal with CD. Next, let's move to the approach, our approach part. As we mentioned, CD is an autoimmune problem. The pathogen is the fragment of 33 amino acid strain remains from the gluten digestion. With high content of the prolyl and the glutamine in this fragment, under enzyme, our digestive system hence, it cannot be broken completely because the high stability. In celiac groups, after gluten intake, intestine immune system will be overactivated and do damage to intestine cells as the direct reason of this disease. And this 33 amino acid peptide is our aim to overcome CD. So first, I will briefly introduce the basic design of our project. We have completed our plasmid design in May, and recent experiments have involved protein expression to test the protein expression and the functional acids for delivery, adhesion, and the release module. They are the three key modules in our whole project design. The final security control section is planned to be refined in September, and we will only draw a draft for it after the probiotics selections. The first module is the delivery. After simply mixing the bacteria with the PV material, we wrap up the engineered bacteria with an enteric coated capsule, which can protect the bacteria from acid damage in environment with the pH of less than 5. As soon as the pH rises above 6, the protective layers distinguished and then release the engineered bacteria. So here comes the second module of our project. That is the adhesion module. In order to overcome the acid-based change in humans' GI tract, we managed to use acid sensing promoters (PASR) to conditionally express the proteins we need. Here are the two recombinant genes that we are designed, in which the CFGA and GS linkers together form the bridge between adhesion molecules and the bacteria in order to provide the space for adhesive proteins CP90K and MEFP5 to function. In this module, we designed two levels of experiment to examine the effect of the adhesion protein. The first is to observe the adhesion effect of bacteria on the slide through the adhesion proteins, on which we will apply special proteins secreted from the small intestine epithelial cells, such as mucus and so on. Here, BSA is only used as a control group. The aim is to simply detect the function of adhesion proteins first. Then the second level is the level of cell. We use the cockatiel cell line to stimulate epithelial cells for intestine, and by counting and analyzing the adhesion of bacteria on the cell layer, we obtain the bacteria adhesion effect result. As for the release module, we add NO sensors as promoters to regulate FVP and PEP. Those two enzymes are all the gluten digestive endopeptidases enzyme. To regulate these two enzymes expression in the intestinal inflammatory environment of the celiac disease patients. NO sensors are chosen as the promoter due to the different concentration of intestinal NO concentration between CD patient and normal people. The NO sensor induced two core components, which are the NO promoters and the operon. They can flexibly regulate the endopeptidase genes expression according to the environmental changes. And the endopeptidase can be released in the environment and function by cutting gluten, and especially for the 33 amino acid peptide. And that process is shown in the diagram. For the enzyme kinetic assays, we use the gluten G12 test kits, which are the ready-to-use ELISAs that detect the gluten in our samples. The G12 antibody targets a 33 amino acid peptide, which is resistant to enzymatic digestion and heat denaturation, and is a fragment of the gliadin protein to which celiac disease sufferers react, making it reliable analytical markers. Listed here are the plasmids used in our experiment, and by comparing the expression levels of these plasmids, we will screen out two plasmids that fit the project expectations. At last, there is a safety control module to be completed in the future, just in case that over amount of bacteria will do harm to patients' intestine. We want to control the added value of bacteria using the principle of quorum question. Once the number of bacteria exceeds our selected criteria, the suicide system is activated for the safe control purpose. And this part is the current experimental progress we do. Until now, the adhesion module has been performed, and the fluorescence detection, pH gradient detection have also completed, and other parts of wet lab work are still in the progress of plasmid fragment construction, because some of our materials we order from other companies just arrived. And now let's invite another team member of IT department to introduce the modeling part.
Okay, this is Astrid. I will introduce our modeling part. Our modeling part has two parts. The first part is the digital simulation of small intestine environment and detect the minimum concentration of bacteria. As the picture shows, our model designs from A to D. We need to impose the initial concentration, which is the concentration of bacteria delivered into the gut with the entering capsule. And after the bacteria released from the capsule, it will release the adhesion protein to help it colonize in the duodenum. Although we have the adhesion protein to help it adhere in the gut, it will also have some loss, so we need to model and quantify this process. And after adhesion, it will release the endopeptidases to digest the gliadin. And in this process, we need to build a comprehensive model which combines the bacterial growth model and a non-kinetic model. And ultimately, we will mix the two models to calculate the minimal concentration of bacteria required to relieve the inflammation. And in this picture, we will write some classical parameters we need to use in our project, such as the pH value and nitric oxide concentration. And changes in these parameters, it will cause effect on important processes such as bacterial growth and protein secretion. And then I will introduce the second part of our modeling. And our second modeling will mainly focus on the selection of probiotics. And our current model uses least absolute shrinkage and selection operator. And in this model, we will consider the parameters such as whether it exists some terrible competition with original gut flora, and its growth rate of the probiotics we choose. And our goal of this modeling part is to ensure the overall stability of the small intestine ecology, because our probiotics predominantly were located in the duodenum, where bacterial concentration and species are lower than other GI environment. And otherwise, we will screen out some probiotics that can help the human body digest the gliadin. Okay, let's go back to the page.
I am back to present the human practice part. For the construction of our HP work, we have three main directions: outreach, IFP, education, and artwork. As our strategy of outreach is point to line, line to face, get a feedback and interaction. From June, we start our public relation work with at least 10 iGEM teams over China and other countries. And through this series of point-to-point work, we get those achievements with other iGEM teams. After getting the object of interaction between team and team, the multi-team and geographic interaction is one of our way to realize line-to-face, such as the network of Greater Bay Area collaboration, co-host education, and allies with similar topics. By the way, symposium-based communication is also an important way for us to change ideas and wisdoms from Conference of China iGEM community to today's Hong Kong-Macau Symposium. We have get vast of feedback and some excellent experience from other teams all over the world. As a part of our IHP work, together with the collaboration and networking, the interaction with companies for industry, academia, research, communication, and the exchange idea with professionals from many different domains are also our important part to get feedback around our project. As one of our three directions to strike golden medals, our education work is a well-designed system demonstrating inclusivity and making our wisdom of synthetic biology accessible to wider population. From special population like Down syndrome in Macau to undergraduate population are all our audience of scientific education. Of course, we adopt very different content and means from different groups of people. And we also make it possible for our education to overcome geographic difficulty and go to more remote areas by co-organizing educational activities with other teams and cooperating with some missionary team in mainland of China. Also, expect offline education. Social medias like WeChat account and Instagram of UM-Macau is also the online platform of our publication, and we have put forward many informations about our project. And so this is all of our presentation, and so thank you very much for your listening, and these are the do you have any questions? Feel free to ask. Okay, so thank you for your listening.